All right, welcome back to Anatomy and Physiology here at Arizona Western College with your professor, Dr. Sean Clavel. Today we're talking about organic molecules, and particularly we're talking about proteins. So, proteins are really like the most abundant and diverse organic molecule in the body. They're responsible for really giving, let's see, where we go. They're responsible for really giving each cell its uniqueness, right? Like the cells are unique in the fact that a lot of the proteins, a lot of the structures that are associated with them are made up by proteins, receptors, channels, the structural support is all primarily made up of proteins. And so proteins are substances that contain carbon, oxygen, hydrogen, nitrogen, and a few other substances like sulfur or phosphorus. They're made by combining different combinations of 20 different naturally occurring amino acids. Sometimes I think of these 20 amino acids as the letters, right? Like they're like your letters of the alphabet. And then the polymers, the actual proteins, are, paragraphs, sentences, pages that are made up by these 20 letters that we would identify as the basic amino acids. And so these amino acids are linked together by the removal of water by dehydration synthesis, and then they can be broken down by the insertion of water, which is called hydrolysis. This is these proteins are held together by, or the amino acids, the monomers are held together by what's called a peptide bond. And proteins in the body include examples like keratin in the skin, hair, and nails. It helps the protein, one of the more common proteins in the keratin is the collagen fibers, which is a protein structure that gives it its strength and durability. The skin and hair are strength and durability. And then another structure that we'll see is hemoglobin, which is responsible for the transport of oxygen through the red blood cells. So, hemoglobin is also a protein structure that we'll identify as well. Now, an amino acid is the monomer of the protein. It's a molecule composed of an amino group, a carboxyl group, and together with a variable side chain. So there's a central carbon located in this. I'm zooming in for you guys. So the primary structure of the amino acid is that there's a central carbon, and then on one side there's a amine. The amine group is the basic. It means it is a proton acceptor. The carboxyl group is acidic, meaning it will readily give its hydrogen. So it's a hydrogen or proton donor. And then the each amino acid is unique in its variable side chain. That's this R side chain here, and that's what kind of gives each of those 20 unique amino acids that make up all of the proteins in our body. Its individuality, for lack of a better term, there. So again, the structure of an amino acid is the central carbon here. There's a hydrogen at the top, so there's a hydrogen atom at the top, but primarily I see that there's an amino group, a carboxyl group, and what we have is oh, a peptide bond, which is formed when amino acids are formed via dehydration synthesis. So strands contain fewer than about 1,000 amino acids are generally referred to as polypeptides, but a protein is made up of thousands or even more amino acids in a chain. And so that kind of brings us to the structure of these proteins, because really the structure of the proteins are what gives it its unique shape. Excuse me, the shape is really what gives it its uniqueness, right? And its ability to function. So it starts with excuse me, let me go back. There's four structures. We'll take the example of hemoglobin, for example, here. There's four structures that we should be able to identify as it relates to protein. And so the first is the primary structure, which is just basically the amino acids, a long chain of amino acids that would be identified as the primary structure of a protein. Again, this may be even referred to as a polypeptide, even as a as opposed to a protein. But the first structure, all of the amino acids held together by peptide bonds. The secondary structure of a protein refers to this folding, where it starts to bend or fold. So you'll see a twisting, an alpha helical structure, kind of this twisting, and this is where hydrogen bonds. Remember hydrogen bonds from our chapter 2? These are kind of weaker bonds that hold together and break fairly easily, but start to give structure, a more compact structure to the protein here. So you can have excuse me, you can have a twisted structure as you see there on the left hand of the screen, or maybe even like a folded sheet structure on the right hand side of the screen. So this is the secondary structure of a protein, when the polypeptide chain is coiled into a helix or pleated into sheets with hydrogen bonds between them. When the hydrogen bonds between the turns are folded to hold them in place. Sometimes the form is held together by a disulfide bridge, but don't worry about that. The tertiary structure of a protein is when the primary structure is folded to expose certain amino acids, while other amino acids kind of fold inward. Hydrogen bonds hold this structure together as well. So this is when the actual, this is when the variable side chains start to interact with each other. So the proteins actually interact, and what we get is this supercoiling. We start to have a little bit of this turns into a little bit more of a three-dimensional structure. So the tertiary structure of the slide, on the slide here, shows the alpha helixes of a heme unit. And this is when the protein begins to exhibit more of its three-dimensional shape, as I mentioned. The model is demonstrating the beginning forms of a hemoglobin molecule. Now, finally, the quaternary structure of a polypeptide is the combining of four or more proteins to form a more complex structure. This level of structure is not very common, and two patterns can develop: either a fibrous structural protein, which we see, like I mentioned, keratin, which is demonstrated in our hair, nail, and skins, or a globular function, which is compact, spherical in shape, and water soluble, which we see here in our example of hemoglobin. And so proteins can be denatured. Denaturing a protein is when the bonds break, holding the folds and the coils. It ultimately changes the physical shape and chemical shape of a protein. Now, there's two primary ways that proteins can be denatured, and the first one is high heat. So heat, again, you can see their body temperature over about 110 degrees. Heat can demonstrate some of those changes. And the example that we often refer to is the irreversible changes of extreme changes that occur when you cook an egg, right? That egg starts kind of liquid, opaque color. And then when it's done cooking, it's a semi-solid structure that is white and unable to look through, right? And so that is an irreversible change to those proteins. So heat is one. The other one is acid or acidity. Acids denature proteins through a change in pH, and this actually, again, changes the physical structure, breaks the bonds between those, and as a result, the shape of that protein is changed and the function of that protein changes. And an example that we often refer to here is that uh, proteins or enzymes are made of proteins, right? So an enzyme is a catalyst. It's a catalyst for it's a protein that provides an environment for a chemical reaction to occur faster than it would in the open environment or faster than it would uh, in our body normally without an enzyme. And the classic example is that these enzymes present as a lock and key. So you can see here the enzyme is the lock, the two particular substrates that'll become the ultimate product are the key. And so each of them join into the lock. And as a result, uh, you can imagine if what would happen if the protein changed its shape, the lock changed its shape, or even the substrates. So if the lock changes or the key changes, if the lock changes or the key changes, then the connection isn't going to happen. And what we have is a chemical reaction that doesn't occur. And so the substrate binding process creates what we see here as a enzyme substrate complex. Let me read this here for you. Enzymes, almost everything that happens in the body does so because an enzyme makes it possible. And enzymes are made of proteins. Enzymes are biologically important because they act as catalysts that accelerate the rate of biochemical reactions by lowering the amount of energy required to start that chemical reaction, right? We talked about how they lower activation energy. Enzymes possess an active site or a groove or a pocket, perhaps, where substrates bind with and undergo chemical reactions. The substrate binding process produces an enzyme substrate complex. This denaturation of an enzyme typically prevents the enzyme substrate complex from forming, and so the chemical reaction shuts down. So that wraps up our chapter, our section on proteins. So remember that proteins, the monomer of a protein is the amino acid. The polymer of a protein is the protein or a polypeptide chain. And so the shape of the protein is very specific to its function. And there are 4 stages of protein structure or function. In the next section, we'll talk about nucleic acids, the ATP, the DNA, and RNA. So I look forward to hearing from you then. Take care and stay safe.